So thank you for the introduction. Okay, so yeah, so today we talk about optical diffraction tomography and fluorescence localization microscopy and how we can merge both together. So this is a joint work with Tan and Pham from who is now at MIT, Ferrol Soulez, who is now at Univ Lyon, and Michael Unzer at the PFL. And the common link between all of us is that at some point, Tanan was a PhD in the group of Michael, and Ferrol and me was postdoc in this group too. So here is the outline of the presentation. I will first present the motivation and the principle of the what I will call ODT SMLM for the merge of the two approaches, the reconstruction method experiment, and then I will give some point about the forward model computation. So let's start with the motivation and principle, and for that I will describe the two methods. So what is a single molecule localization microscopy? We can find it in two forms, which is also known as Palm and Storm. I will not enter into this detail. This is the way we implement it in practice. But the main principle behind this is that we have like a fluorescent molecule that have different states, and they can be, as we see here, in non-excited state. Then we can activate just a few of them, and once they have been activated, we can localize them, and the cross means that the one that have been localized. So it's like instead of doing one image, we take several images, and each time we just activate a sparse subset of fluorophore, and like that we can reach like a resolution of the order of 10 nanometers, around 10 nanometers. So here it's an example with images, so we can see like the images of the activation, the localization, and the difference between what we get with this technique and a standard wide field image. So this is in 2D. We can also get 3D information with that. And for this, we use, I would say, exotic point spread function. For example, astigmatism. So here it deforms like vertically and horizontally depending on the Z position, or the double helix that the two lobes here turns around each other depending the Z position, or the biplane modality is like we take the acquisition at two different planes, and we can see different defocus that is related to the Z position. So on the other hand, I will be interested in optical diffraction tomography, and here the principle is not to reconstruct the fluorescence signal. There is no fluorescence. We try to reconstruct the refractive index of the sample, and for that we will record several images that correspond to the diffraction pattern of the sample when we send plane wave, so with different orientation. So we have the incident field that we send to the sample, it's scatter, and we record the total field. So we can record the total field, a complex measure of total field through holography system. But in this talk, it will be phaseless optical diffraction tomography in the sense that we will not have the phase information. And then we have the inverse scattering, is that from this measurement, we want to recover the refractive index of the sample. So from these two setups, on the one hand, we have the functional information with a single molecule localization microscopy, because we can put the fluorescence probes into the structure we are interested in. And on the other hand, we have structural information from ODT. So the goal here is to get these two information from the same data. And on the other hand, having access to the refractive index when we estimate it will allow us to take into account in the estimation of the molecule localization the aberration that are induced by the sample itself. The general principle is to consider here that the molecule inside the sample act as the illumination source, and they travel through the sample, diffract, and will be recorded. So here we will consider the biplane modality, and we have this intensity measurement. So the goal will be to jointly reconstruct both the refractive index and the molecule localization, molecule position and amplitude from the sole SMLM acquisition. So there is few related work. The first is that there have been some systems that have been designed to acquire both fluorescence and phase data. So in this case, you need a complex setup, and you have these two kinds of information. The brief method that has been proposed is very similar to what I will present here, except that the way of acquiring the data is a bit different. It's not related with the goal of exploiting SMLM data in another way. It's a specific system that is built to have this kind of illumination. And the last one is for SMLM data, but in this work, the author consider like we can record complex measurement, which is not the case in practice, and they also use a simplified version of the model. So I will now go into the reconstruction method we consider. So first of all, the model for the emission of a fluorophore at a certain position with a certain amplitude, we model it as a spherical wave, okay, which has this expression. And then the image formation process that we consider is as this form. So here, what we see first the UT is the total field, that is the interference, I mean the scattered field from the sample plus the incident field. And so I will discuss a bit this total field later. So we consider a Poisson noise model. We have also a background fluorescence that is modeled here. We have the camera sampling points, and the P here models the optical system. So typically it models the objective. Concerning the total field, it is governed by the Lippmann-Schwinger equation, which is the integral form of the Helmholtz equation that models wave propagation. And here we can see two main components. It's the green function and the scattering potential, which is related to the refractive index. Okay, so in the following, the F will be related to this refractive index. So one difficulty of this model is that it is nonlinear in F. The quantity we want to estimate here is F. The data is U, T. And so we see that the relationship between the two is nonlinear. Okay, and but okay, it is nonlinear, so more complex to handle, but it handles multiple scattering, and this will allow us to have a more accurate reconstruction. So from this model, we deploy a joint optimization problem, which is composed as usually of two terms, one data fidelity term and one regularizer. The data fidelity term is made of the Kullback-Leibler generalized divergence that accounts for Poisson noise. And for the regularizer, we consider the total variation regularizer. And we want to optimize with respect to both the position of the fluorophore, their amplitude, and also the refractive index F, so which is a discrete version of the refractive index. So for that, we'll consider an alternating optimization. For what concerns the estimation of the amplitude, so we can use the fact that actually when we change the amplitude, we can write it in this way. So for that, we will have a simple expression of the first and second derivative, and we can derive an efficient Newton update. Concerning the update of the position of the molecule, so here we also want to derive a gradient-based method, but the problem we faced was that the incident field, so the emission of one fluorophore, this spherical wave, actually was not differentiable. So we smooth the norm that by adding here a small epsilon to make it differentiable, and we can have a closed form expression of the gradient and deploy a gradient descent here. And for the update on F, for that we need, it consists of a phaseless inverse scattering problem that we solve using a relaxed version of the FISTA algorithm. And for that, it requires to have the Jacobian of our model. So this, we did that. We have an efficient way of computing it, and we did that in the previous work. And we also need the proximity operator of the of the TV, and here we deploy the fast gradient projection algorithm for this step. So before to go into the experiment, we are missing some steps. The first one is about the background. So the background we estimate it at the beginning and we don't touch it. And for that, we assume that it varies slowly, both in space and time, because we have several acquisitions in SMLM. So this corresponds to the different acquisition in time. And for that, we use a simple two-step method where we'll mask the detected molecule around them. And then we use a spatial temporal median filter in order to paint. And this was making the job for that, this simple method. And concerning the initialization, as the problem we are trying to solve is highly non-convex, so we need to try to have good initialization. So concerning the initialization for position and amplitude, we can use any single molecule localization microscopy software. Okay, in this case, we adopted a simple cross-correlation based approach. And concerning the refractive index map, so usually in phaseless ODT, we can, for example, use a simple, an efficient method that is light field refocusing. The problem is that it requires to have coherent light source of known geometry, and we don't have this here. So, okay, we simply take the wide field image, replicate it along Z, and smooth it to have a very coarse initialization of the refractive index. So I will now show you some experiment we did in this context. So for that, we simulated this volume. So in the left, it's the refractive index volume of the cell we simulated. Inside it, we put some kind of compartment with it can like cell within cell, and we will populate this kind of membrane with fluorophore. And then we simulate our SMLM acquisition. Okay, in the bottom it's the corresponding wide field image, and here it's the biplane modality for SMLM. So each time we see two different planes that are recorded, and we can see that okay, we are in an extremely low density regime where more or less we activate only one molecule at a time. And yeah, we generate also a background that is varying slowly both in space and time. So this is the setup we considered. And here are some results concerning the refractive index estimation. So on the first column, it's the true volume. And the second one is the initial guess for the refractive index. So as I told you, it's a very coarse estimation. It's just a replication of the wide field image that is smooth. The third column, for this one, we keep the estimation of the position and amplitude of the molecule, the initial one, and we don't optimize them. We just optimize the refractive index given this position. And we see that it did not work at all. The fourth column is correspond to the joint optimization. And the last one is when we fix the position and amplitude of the molecule to their true position, and we just optimize. So it's what we say, what we would like to get with our joint optimization. So we can see that we are close to that. Even if we have some artifact for plane that, for example, minus 0.2 micrometer. In this plane, we can maybe, I'm not sure it's very clear but we can see some artifacts that are due, and I will come back to that later, to some position and amplitude of molecules that have been not precisely estimated. And to try to explain for that, the illumination source here in this setup are within the sample, because there are fluorescent probes that are within the sample. And we can try here to illustrate what is the region of the sample that a molecule will provide information about. And we see that like the Z position, the negative Z position. Okay, here we have the direction for the Z axis. We'll have much less number of measurements that contain data, information about this part of the sample. Okay, so this is one reason why we reconstruct better in for Z equal 0.2 than for Z equal minus 0.2.
You have 4 minutes in total.
Okay. So this is the estimation of the corresponding estimation of the molecule position and amplitude. So at top, it's the initial guess. So it's a projection on the YZ axis. So we can see that our initial estimation is on the grid. Okay, but then the refinement is off the grid. And again, we have the comparison when between the ground truth and the joint optimization. And we can see here the improvement we get with respect to the initial position and amplitude. And also we have the comparison when we consider the initial F0, so refractive index, the initial refractive index, and we just optimize the optimization, or if we fix the refractive index to the ground truth and we optimize the position. So we see that given this information of the refractive index that have been estimated, we can also improve the localization of the molecule. So we wanted also to test the effect of the fluorophore distribution. And here we put like all fluorescent molecule within a region that, for example, can be the nucleus. And we clearly see, and this was expected, that we don't reconstruct as good as in the more, we say, uniform case. And this is explained by the illustration I showed you before. Okay, because we don't have enough information about the totality of the sample, and we construct more precisely the center part here, that where there are the molecule. And finally, we tested the robustness to noise. Okay, so here the error parameter controls the noise. When it is small, there is higher noise. When it is large, okay. And in the bottom, we have some example of data. So we see that it is quite robust to noise. And here we have box plot with in red the initial error on the position and amplitude, and same for in green is after optimization. So we see that it remains outlier. Okay, most of them most of the error have been reduced, but we can see that some of them have been enlarged, and this is because our initial estimation of the refractive index is very poor, and some give the first optimization we do for the position and amplitude, it can move away, and we should be able to correct for that, to detect this one and reinitialize them, for example. I mean, we didn't have a look to that. So maybe I have a few minutes. I will just quickly go over this four model computation, just to maybe show you the difficulty of that. So as I was saying before, in the four model, we need to compute this integral that involves the green function. So we do it in two steps. We first compute the total field into the region which contains the sample. And given this total field in this region omega, we can compute the field on the region gamma, which is the detector, and this improves speed and reduce the memory consumption. So the second part is what about the discretization of the green function? The problem is that the green function is both singular in spatial and Fourier domain, and to deal with that, we use a truncated version of the green function. Okay, because as the sample we look at is compactly supported, the convolution remains unchanged if we truncate the green function in the spatial domain. And the interest of that is that in Fourier domain now we have a smooth function that we can sample properly. So in red here in the 1D plot, it's the initial green function is Fourier domain, and the complex in imaginary part in blue and green are the same for the truncated version. And for that, we can compute efficiently this using FFTs and have a proper discretization. Okay, I will just show this. This is a comparison between the computation using this truncated version of the green function or a naive approach where we just select a discretization of the green function by taking grid points that does not contain the singularity. And we can see that we can, n is the number of sampling points. Okay, so we can see that the error in a case where we know closed form expression of the result of the convolution, that we can reach machine precision if we have enough sampling point compared to the naive approach. Okay, and this was for like reducing time and memory, but we can discuss of that later. So to conclude here at the top, you have like the main paper which is related this work, and the third one. I mean, the third line here corresponds to the where we describe this computation of the forward model. Everything has been implemented using the global BioIM library. And finally, I would like just to advertise there is a postdoc position that is open in Toulouse with Pierre Weiss. It will be at the Centre de Biologie Intégrative de Toulouse and start in fall 2023. So if you are interested, don't hesitate to contact Pierre or me. So thank you.
Thank you very much, Emmanuel, for this presentation. Do we have a question from the chat? No. Is there any quick question from the local audience? This one, Roland.
Thank you. Very interesting talk. So as far as I understood, you have to activate the molecules one at a time, right?
Yeah.
Could you activate them by group, or is it a problem, or would you need to assume that they reproduce incoherent lights to do so?
Actually, for the moment, for simplicity, we assume that we have activate one at a time. In practice, this means that we need to be in a very low regime. So, and you don't control exactly the number of molecules you activate at each iteration, but depending on the power of the laser you use, you can have like probabilities that there is few that are activated. But we can easily extend the model because everything is there are incoherent source. So the model, if we have 2 or 3, will be the sum of all of them. So we didn't implement that yet, but yeah, it's not that hard to do.
Okay, thank you.